Alright, welcome everyone. My name is Kevin Libwood, and this is another video for our series of training materials for Theagen's Terra workflows for SARS-CoV-2 genomic analysis. Today's video is part 1 of 2 for understanding outputs from the Titan workflows for genomic characterization. And in part 1, I'll be focusing on defining QC metrics made available by these workflows. And in part 2, I'll be discussing clade and lineage outputs that are also generated. Alright, so with that, let's get into it. And just as a refresher, the Titan workflows for genomic characterization include 4 separate workflows that take sequencing data from 4 different sequencing approaches: Clear Labs read data, ONT data, and both paired N and single N Illumina read data. And with input reads from these platforms, these Titan workflows generate SARS-CoV-2 consensus assemblies, provide metrics to gauge the qualities of those assemblies, and provide some genomic characterization through pangolin and nextclade typing. And this genome assembly is pretty self-explanatory. This is the consensus genome assembly in FASTA file format that is generated from these workflows. But the QC and lineage or clade output may be new to a lot of users. And so the focus of these next 2 videos are on understanding these outputs. For the quality control metrics, I hope to provide some foundational information regarding the definitions of these metrics, outline some of the guidance thresholds that have developed throughout the public health community, and highlight how some of these metrics can be utilized to troubleshoot wet lab issues that may be influencing the quality of read data. And as I mentioned, I'll be dedicating a separate video to dive deeper into the lineage and clade typing output generated by these workflows. But for this video, let's hop into these QC metrics. Okay, so at first broad view, I like to categorize the QC metrics generated by these workflows into 3 categories. First is a pass warning level to quickly identify the high quality assemblies and determine which samples may require a closer look to determine the confidence in some of the reportable outputs. Next are these troubleshooting metrics. So for samples that don't get that first level pass, what metrics can we start looking at to start investigating what may have led to poor first level metrics, and if there's any kind of corrective action that can be taken on the wet lab side of things to address these issues. And then the third level of things come by way of digging into the intermediate files generated through the assembly process. Now in this introductory video series, I won't be able to cover all the background and nuances necessary to perform these deep dives, but I do want to ensure that users are aware of which files are available to them if they ever care to investigate these themselves, or perhaps have access to other collaborators such as academic partners that can look at these files more closely with them. Alright, so with that, let's take a look at these first level pass warning metrics. So to understand the first level look, I think it helps to contextualize what the ideal spirit of this sequencing experiment is. So if we picture the full SARS-CoV-2 genome in your sample here in blue, the ideal sequencing experiment will allow us to take this biological molecule, do all of the wet lab protocols to generate sequence data and all the bioinformatics to assemble it, and at the end of it all, we get a full and complete consensus assembly that represents the entirety of the biological molecule in our sample. Of course, we rarely hit this mark, and instead we generate a fragmented representation of the original SARS-CoV-2 genome in your wet lab sample. So what these first level metrics try to gauge is how fractioned our consensus genome assembly is relative to the original SARS-CoV-2 sample sequence. And all 3 of these metrics are a different way to assess that. First, we're looking at the assembly length unambiguous. This figure is the total count of unambiguous bases, that is A's, T's, C's, and G's, within the final consensus assembly. So a higher value in the assembly length unambiguous is an indicator of a stronger consensus assembly. And so the sort of inverse of this figure is the number of N's, or the number of ambiguous bases within the consensus assembly. And it's worth noting that there are varying levels of ambiguity. For example, there may be positions in the assembly at which the consensus assembler couldn't decide between one of 2 nucleotides. So rather than an N, there are IUPAC codes such as W, which represent either an adenine or a thymine at that position. In this context, however, we're speaking about N positions, which are loci that are fully ambiguous because of either too little signal or too much noise in the sequencing data to make a confident call towards any nucleotide. So the higher number of N's, the more fragmented your consensus assembly is. In short, a lower number of N's is an indicator of a stronger consensus assembly. And then the third metric here is the percent reference coverage. This is essentially taking that assembly length unambiguous metric to gauge how much of the original Wuhan 1 reference genome that our consensus assembly covers. So to calculate this, we take the assembly length and ambiguous and divide this by the known length of the Wuhan 1 reference, which is 29,903 base pairs, and convert this into a percentage. So in an ideal experiment, this value will be 100%. That is, we were able to cover with our consensus assembly 100% of the original Wuhan 1 SARS-CoV-2 reference genome. In short, a higher percentage reference coverage is an indicator of a stronger consensus assembly. Okay, so in looking at all of these metrics, here are some suggested guidance thresholds that you might use to determine whether or not your sample passes your initial QC check or flags some kind of warning that would initiate further investigation. So an assembly length and ambiguous of more than 24,000 base pairs, fewer than 5,000 ends, and a percent reference coverage of above 83%. And these metrics here are based on various database standards and communications in the public health community. GISAID, for example, won't accept genome submissions for samples with greater than 5,000 ends. Nonetheless, these are simply suggested guidance thresholds. The thresholds labs choose to adopt for their internal QC protocols are fully up to the laboratory's discretion. All right, so let's move on to the troubleshooting metrics. If you run into a sample that gets flagged with a warning with these first level metrics, these next QC outputs can help you determine what may have gone awry in the wet lab process to identify issues of potential corrective action. And so to understand these next metrics, it will be important to have some context in how we go from the original SARS-CoV-2 genome to creating this consensus assembly. So in a huge broad stroke, here are the 3 basic steps to go from SARS-CoV-2 sample to a SARS-CoV-2 consensus assembly. The first step is the PCR tiling process with the Arctic v3 protocol. And in this protocol, 2 PCR reactions are performed in 2 separate pools and then combined before sequencing. And the next step is the actual sequencing of these amplicons. And then the third step is the consensus assembly process, mapping these amplicon read data to the Wuhan 1 reference to create the consensus assembly. And for each of these steps, the Titan workflows for genomic characterization generate QC outputs that give insight on how well each of these processes went. In looking at the 2 separate PCR reactions, we can see in silico how much read data was generated from each pool. And these data are conveyed in the pool 1 and pool 2 percentages. What will sometimes happen during the wet lab portion of sequencing is that a PCR reaction will fail. So these metrics here will help you identify if that was the case. It's also worth noting that this metric is only available through the Titan Clear Labs and Titan ONT workflows, since both of these pipelines utilize the Arctic Medaka protocol for assembly, which makes access to these numbers incredibly straightforward. Titan Illumina paired N and Titan Illumina single N, unfortunately, perform consensus assembly through a different methodology, and calculating these values will take some level of additional development before we can grab these metrics as a default output. Okay, so next is the process of actually sequencing these SARS-CoV-2 amplicons with your NGS platform. And in this step, ideally, you'll be able to generate loads of read data for specifically the SARS-CoV-2 amplicons you generate. So the first thing you can check to see if this is true is to see plainly how much read data was generated by looking at the FastQC raw metric. This is the total count of read data within the user-provided FastQ file. So the higher that this count is, the more read data was actually created during the sequencing experiment itself. And then this next metric is to gauge how much SARS-CoV-2 and human data is represented in your raw read data. Because of the sample types we're working with and the method of sequencing, it's likely that some level of human DNA will be sequenced. It's incredibly unlikely that this will represent enough human DNA to be determined as PHI. But some human DNA will be present in your sample. So we use the Kraken tool to calculate how much human and SARS-CoV-2 reads are actually present in your sample by percentage. Of course, in an ideal experiment, you want the human read data to be as low as possible and the SARS-CoV-2 percentages to be closer to 100%. Okay, and then now let's look at that third step in this process, the actual read mapping. So in thinking about this process, I think it will be helpful to have a bit of a visualization, and big thanks to Lee Katz for making the first iteration of this little animation that I've over the years modified and changed to fit different training workshops. Okay, so in this animation, imagine that you have a reference genome here in blue, and then with sequencing you generate your read data here in gray. When performing consensus assembly, these read data are first mapped to the reference genome. All of the read data stacked at each position are compared to identify the variants and unchanged nucleotides to generate the consensus assembly. So one metric we can look at to gauge this read mapping experiment is to look at, on average, how many reads aligned to each position in the reference genome. And so we call this metric the assembly mean coverage. And this can be a bit confusing since we use coverage in a different context to describe how much of the original reference genome was covered by the consensus assembly. In this context, it may be more helpful to think of this as the depth of coverage for the assembly. But in short, the higher your assembly mean coverage, the more SARS-CoV-2 data you have to support the consensus assembly generated. Alright, so in terms of guidance thresholds, your FastQC numbers and assembly mean coverage are going to be very dependent on the wet lab protocols you're using at your laboratory. So if you have questions about your specific protocol and where the guidance thresholds are for these metrics, I'd recommend reaching out to either the manufacturer or other laboratories that are using the same sequencing protocol to find out what the minimum acceptable values are. For the crack in human scores, we've seen a bit of discrepancy in the ONT and Illumina data, with rough averages of 5% of human DNA showing up in ONT data and 15% in Illumina data. Anything higher than this may be an indication of too low volume of SARS-CoV-2 read data in your original sample to generate a proper assembly, and or an issue somewhere in the wet lab protocol that is leading to so few SARS-CoV-2 reads being generated. And for the pool 1 and pool 2 representation, you really want to see ratios close to a 40 to 60% representation in either direction. Once you see numbers skewed to 1090 or 2080, that's a strong indication that one of your PCR reactions failed and that resequencing is likely required to capture a strong consensus assembly for that sample. Okay, and the last level of quality metrics for these workflows is diving deep into the actual intermediate files generated by the Titan genomic characterization workflows. And again, this is meant to be an introductory overview of these metrics, so I won't have the time to get into the nitty-gritty nuances of these files, as the process of analyzing these types of files usually requires some level of manual assessment. But I did want to mention what files are available for this kind of assessment. And so the first one we're going to look at is the binary alignment map file, or BAM file. This file is generated when performing the actual alignment itself and contains all of the information regarding read alignment to the Wuhan 1 reference. And these files can be visually inspected by commercial software such as Genius or CLC Workbench, or by free software such as IGV, the Interactive Genomics Viewer. And here's an example of what some ONT data looks like in IGV. You can see on the bottom part here is the actual read data mapped along the Wuhan 1 reference. And the other intermediate file that can be incredibly useful when digging deeper into the quality of a consensus assembly is the variant call format file, or VCF file. This is a text file that contains all of the relevant information regarding variants identified in the consensus assembly. The most common format is VCF, but a TSV that contains exactly the same information is output by the Titan Illumina paired end and single end workflows generated by IVAR. For those of you who want to learn more about these files and how to interpret them, I will link some resources below which include a great walkthrough video of interpreting SARS-CoV-2 BAM files posted by Ali Black and CZI. Alright, so that's everything I have in this introduction to QC outputs. In part 2 of understanding Titan genomic characterization workflow outputs, I'll be covering the next clade and next strain outputs generated. But that's all I have for now. I hope this was clear for everyone and that you were able to learn a little bit from this video. Again, if you have any questions, please feel free to leave them in the comments below, and we will be sure to clarify as much as we can. Thank you, and I'll see you in the next one.